Welcome to episode, and the topic of our discussion is pulse field gel electrophoresis. Pulse field gel electrophoresis is actually a modification of the normal gel electrophoresis. So here is a picture of a standard or normal gel electrophoresis. So we use the gel electrophoresis for the separation of the DNA fragments. Now DNA is already negatively charged, and so it moves from this cathode to anode in this direction. So whenever we used the DNA fragments of size greater than 40 kb, so the separation is not done because the fragments which are greater than 40 kb, they move together independent of their size, and as a result the separation is hindered. To solve this problem, PFGE was developed. Now in PFGE we use the electric fields which switch periodically, unlike the normal gel electrophoresis where only in one direction the electric field is applied. So we will learn about this PFGE and how the sample is prepared and the principle in this video. So here are the modifications which are actually done to prepare the PFGE. So we used 0.8 to 1% of agarose while making the gel. Now since it is a very small amount, so the pore size will be bigger, and since the pore size will be bigger, the larger fragments will be separated easily. Next, the direction of the electric field is also switched periodically during the process of separation, and the various length of the DNA will behave differently at different rates depending on the periodic change of the electric field. So the first electric field is applied in this way, like in this direction. The next is applied in this way, and another one is applied in this way. So all electric fields are applied from this negative to positive in 3 directions, and these directions switch periodically, and this switching actually changes the behavior or the direction in which the DNA fragments are moving, and as a result the separation becomes efficient. The next is we use lower voltage. Low voltage of about 6 volt is used, and why it is used we will learn in this video. So here we have a detailed process, and at first we will start with how we can prepare the sample. So here we have the cell culture, and we take an inoculation loop and we take a loop full of it and put it in molten agar, and we take the molten agar in a syringe and let it solidify, and we can see that this small part coming out, this is the solidified agarose gel which contains the cell culture in it. And we will use a very sharp razor and we will cut a slice of this gel, and this will be called as the plug. Now these plugs, we apply the lysis buffer. This lysis buffer is used to lyse the cell and extract the DNA. Once the DNA gets extracted, the restriction into nucleus is also added to the plugs in order to cleave the DNA. So you see, this is a plug which is very small. You can zoom it to see. So this plug is actually taken in, cut, and they are put in these wells like this. So this is a schematic representation of how the plugs are put. So we place the plugs like this, and now see, this part is empty, right? So in this part we actually pour the molten agarose in order to seal this entire thing and make sure that no air bubbles is present. So once they are sealed, then they are good to go. And remember that the molten agarose should be at room temperature while you are putting it in the well to seal it. And the next thing is, the size of the plug should be 5 to 10 microliter. Now since we are using the syringe, we can easily understand that the 5 to 10 microliter, that how much we are extracting. So it should be very thin. It's a very thin piece because it will help the DNA to diffuse quickly from the plug to the gel. That's why. So this is the entire setup of the PFGE, and this hexagon is actually the electric field, and inside this electric field we are placing our gel. This is the gel where the plugs are also inserted, and we place it in the electric field. And as I said, the direction of the electric field will be like this, in one direction, and then it is from this side, and another one will be from this side, and the entire angle is 120 degree between this and this field. Now here's one concept. So whenever we have a small fragment of DNA, to linearize it, a very small time is needed compared to the ones that are bigger. For bigger fragments, to linearize to elongate the fragment, more time is needed. Now we will go back to this picture. So this is a gel of normal gel electrophoresis, and here the path length is straight and it's same for all the fragments. So let's say we have a mixture of DNA fragments, like it has this, and all these three types of fragments are mixed together. Now for separating, to elongate this fragment, this smaller fragment, very less time is needed, but in the time limit is same for all the fragments. So this separation will be done efficiently, but the separation of the bigger fragments will not be done. So that is why the pulse field gel electrophoresis is used. Here what happens, the DNA fragments, they move in a zigzag pattern like this way, and this zigzag path is done by the electric field. So this electric field moves the fragment in such a way that they form a zigzag path, and you can imagine it in this way. If we linearize this zigzag path, if we straighten this zigzag path, then the length will be much more compared to this straight path. Although the size of the gel is more or less same, but still the path length will be more for the PFGE compared to the normal gel electrophoresis, because it is moving in a zigzag path. The smaller fragments that are present in the mixture, they will quickly align compared to the larger DNA fragments, which will take much longer time to align. So that is why by increasing the path length, we are increasing the time for which the separation is done, and as a result, both the smaller and the larger fragments, they will be separated in the gel. So here we have this zigzag path, and to understand why it is happening this zigzag pathway, let's say the electric field is applied in this direction. Follow this laser arrow. So in this direction the electric field is applied, that is towards the right, and during this time when the electric field is applied to the right direction, the direction of the fragment is in this way. So see this line, just this line is directed in the right. So next, when in this junctional point, when it reaches this point, the switching of the electric field is done. So now the electric field will move in this way, like this, that is towards the left, and as it is moving in the left, the direction of these fragments, the direction of the fragment will also change, and it will move towards the left. Like this, the separation is done and the zigzag line is formed. I think you can now understand why the zigzag path is formed. It is because of the switching of the electric field. We also mentioned that we are using low voltage of 6 volt. It is because when you are using the low voltage, the motion of the fragments, the motion in this direction, the motion of the fragments will become very slow, and since the separation time is increased, that is the DNA fragments are moving very slowly, so larger fragments, they are very efficiently separated. So that is why we are using low voltage for PFGE. Now here we have a diagram of the results of the PFGE, and it is taken from ResearchGate. So here we have 3 examples, like one taken in 120 seconds, another one is 48 seconds, and another one is 16 seconds. So these are actually the switching times. So when the switching time is high, the separation of the fragments is very efficient, you can see that. But when it is 16 seconds, just 16 seconds, the separation is not very efficient. So we can say for proper separation, efficient separation, we should use a switching time which is very high. That is let's say that is 120 seconds. So what that means is that for 120 seconds, the fragment will move in one direction. Then after 120 seconds, again the fragment will move in a different direction. So this the formation of this entire line will take 120 seconds. This is the switching time. So that is it for today. Thank you for watching and subscribe for more updates.